topic of my discussion is Vermi-technology, utilization of earthworms as progressive sustainable waste management practice. So first of all, I am introduce you the word waste. Waste are the unwanted or unusable materials. Waste is any substance which is discarded after primary use or is worthless, defective, and of no use. And different organizations describe or define the waste in the different ways, as United Nations Environmental Program, United Nations Statistic Division, and the European Union. But all of these, they want to say that the waste are the object which the holders discards, intend to discard, or is required to discard. So there are different types of waste. The physical states of the waste are either solid waste, liquid waste, and gaseous waste. But these waste are either in the form of biodegradable, non-biodegradable, hazardous waste, non-hazardous waste, controlled waste, and non-controlled waste. And these all waste are either garbage, rubbish, ashes, dead animals, industrial waste, mining and agriculture waste, animals, byproducts, biomedical waste, commercial waste, chemical waste, or forest waste. But here I am also showing you the sources of the solid waste. As I discussed above, that we have a controlled waste and uncontrolled waste. Controlled waste released from the household, commercial waste, commercial areas, industrial area, educational or institutes, hospital waste, that is the controlled waste. And uncontrolled waste relate to agriculture waste, mines, and quarries. But today I will discuss about the organic solid waste that released from different household, commercial, industrial, educational, hospital, both controlled and uncontrolled waste. As you know that the waste management concept has a 4 hierarchies, or the 4 R's: reduce, either reuse, recycle, or recover the waste management. And for these we are using collection, transportation, processing, and disposal. After collection, we store the waste material and then transport to the laboratories or other places for the processing, either through mechanically, biologically, or thermal, and then we dispose of. But here is I am showing you the different disposal methods. Landfill technology, aerobic and anaerobic digestion, composting, gasification, recycling, incineration, and pyrolysis. But these all disposal methods emit the greenhouse gases and have a negative impact on the health as well as on the environment. In case of the health, different diseases caused either soil-associated, vector-borne, waterborne, or airborne, or foodborne. On the other hand, the drawbacks of solid waste management systems are have a negative impact on the environment. They emits the greenhouse gases, and due to which the global warming, ozone layer depletion, runaway greenhouse effect, acidification of water bodies, smoke, and air pollution occur, and which lead to the climate change. So what are the overachievement? What we did? We established the Vermi-Tech unit at University of Azad Jammu and Kashmir, Muzaffarabad, under HEC Higher Education Commission Technology Development Fund Program. And we established different Vermi technologies: vermi-composting, vermi-remediation, vermi-filtration. I will explain you these terminologies in the coming slides. And we also developed the integrated sustainable technologies to manage the solid waste. And for this, we are doing vermiculturing, vermicomposting, and vermibacteria. Here I am showing you the logo of the Vermi-Tech unit and our copyright. And here is the detail of the HEC Technology Development Fund project, and worth of 6.757 million. These are the researchers that involve HEC Technology Development Fund and the establishment of Vermi-technologies. Rosina Ghulam Mustafa, PhD student, and she did the work on the diversity of the earthworm species that are used in vermicomposting. Irsa Shafiq produced the vermicompost from the organic waste solid management, and with the help of earthworm gut bacteria that isolated by her. Anam Nasir established a vermi-remediation technology that's used for the remediation of heavy metals from the contaminated land, and using the various earthworm species and other hypoaccumulators like plants. Sadia Yunus produced vermicompost from goat and sheep manure. And Saim Malik checked the effect of vermicompost that released from the solid waste and on the various crops and growth and development. What is the scope of the study? To develop the sustainable agriculture technology to produce nutrient-rich vermicompost that could be used as a organic biofertilizer, boost up the green economy, and reduce the environmental pollution. Here we meet the sustainable development goals, good health and well-being, clean water and sanitation, decent work and economic growth, industry, innovation and infrastructure, sustainable cities and communities, climate action, life on land, and partnerships for the goals. And these all parameters we meet or achieve by this project. What is the worth of the vermicomposting? Why we did vermicomposting? First, we can reduce the usage of agrochemicals that have negative impact on health as well as on environment. And second is it is a better or proper or eco-friendly solid waste treatment and disposal method. And others are mentioned here: organic waste management, nutrient cycling, soil enrichment, reduce the environmental impact, community and urban agriculture, biodiversity promotion, sustainable agriculture, economic benefits, water retention. We can educate and awareness maybe occur in about the vermicomposting in our community, soil erosion prevention, and livelihood opportunities. So here I am describing you what is the Vermi-technology. Vermi is the Latin word which comes worms, and the Vermi-technology is the process of using earthworms, either decompose organic waste material into nutrient-rich compost. We did vermicomposting, and we produce a vermicompost either to remediate the heavy metals and toxic compounds from contaminated sources, and we established vermi-remediation technology, and to purify the wastewater. One of the PhD student working on the vermi filtration, that we are using earthworms we are using earthworms for the purification of water. Why are we using earthworms? Because earthworms used in various disciplines of sciences, either agriculture, environment, pharmaceutical, biomedical, poultry and fish and poultry industry. How? Because they produce different products. Worm meal that used for the fish and poultry industry, animal model, earthworms used as an animal model in various educational and institutional institutes. Vermibacteria used as a plant growth promoting bacteria. Pharmaceutical products of earthworms are also used because they have a potential antibacterial, anti-cancerous, and anti-inflammatory activity. We can use earthworms for the remediation of heavy metals. And vermi wash, vermi tea, or vermicompost are used as an organic biofertilizer to enrich the soil or to improve the soil health after production of solid waste. Here I am introducing you some of the introduction about the earthworms. That the earthworms are the terrestrial invertebrates, originated 600 million years ago in the Precambrian era, have a diverse habitat, belong to phylum Annelida, Chaetopoda, and Oligochaeta, and they are distributed into different ecological strategies based on the soil horizon: epizooics, anisakes, and endozooics. Different earthworm species were used in the vermicomposting: Ferritima lignicola, Ferritima haviana, Ferritima postuma, Esnia foetida, Perionyx excavates, Euryalus eugene, and Esnia andri. And in the current study, we used Esnia foetida for the vermicomposting. At the Vermitech unit, we produced the vermicompost on a small scale. And for the small scale, we used small tubs of 20 kg. And in the 20 kg tubs, we produce a vermicompost by using various material, pre-digested vegetables and fruits as a raw material, cow dung, cattle dung, dip waste papers, coconut peel, dried grasses, earthworms, rice straw, eggshells, and wheat straw were used. And this vermicomposting process is done for the 90 days. And after 90 days, we collect the vermicompost. Vermicompost also produced on the large scale at the Vermi-Tech unit. And here is I am showing you the structure of the vermicomposting process that we are doing in the pits. And the pits are 10 feet and 5 feet. And the different organic waste material are poured into the pits, and we are doing watering, and then we produce the food, fresh food, spinach for the earthworms, and then after 90 days, we sieve the vermicompost. What are the mechanisms behind the vermicomposting? When we add when we add earthworms in the raw material or in organic solid waste, it is a joint action of earthworms and microbes, because in the earthworms, the gut of earthworms also contain the different types of microbes that involve in the degradation of solid waste material, because these microbes are released the hydrolytic enzymes. And during this process, carbon dioxide and heat generated and then absorb the oxygen, and two products are collected from this process. Vermicompost or the vermi wash, and both could be used as a organic fertilizer. After production of vermicompost, we analyze the properties of the cow dung and vermicompost, because different in the feed, the cow dung also used as a biofertilizer. But here we compare that vermicompost shows highest NPK, that is the NPK rich, and also enrich in the micronutrients, and the pH is 7.8, electrical conductivity 2.82, and moisture has a 25%, organic matter 43% as compared to the cow dung. So here is the smell is the earthy smell of the vermicompost as compared to cow dung. So we can say that vermicompost is a better organic fertilizer as compared to the cow dung. After these, why we vermicompost promote? Because it is a good soil conditioner, plant growth promoter, high porosity aeration. Aeration, drainage, and water holding capacity, high level of beneficial soil microorganisms, rich in growth hormones, rich in nutrients, rich in humic acids, free of pathogen, free of toxic chemicals, is a plant protector, and induce biological resistance in plants, because these are due to the presence of earthworms. Because earthworms are the good turners, good mixers, good aerators, screeners, accelerators, and the pathogen controllers. After the production of vermicompost, we check the impact of vermicompost on various ornamental plants and the other crops of the Pakistan. And we observed that for this experiment, we use different concentrations of the vermicompost, and seedling parameters and vegetative growth parameters of marigold and pansy and ornamental plants were checked. And we observed that the 20% vermicompost showed the maximum seedling of the marigold. And here I am showing you the length of the whole plant. And 35% show the maximum vegetative growth parameters of the marigold when we use 35% of vermicompost. On the other hand, 30% of vermicompost shows the maximum seedling of pansy, and 35% shows maximum seedling parameters of the pansy as compared to control. And you can see easily the difference between the control and the different concentrations of the vermicompost. After that, here I am showing you the impact of germination media on marigold after 28 days of seed germination, and difference clear were observed when 20% vermicompost was used. In case of marigold, pansy, 28 days of seed germination. We also checked the impact of vermicompost on seed germination and seedling growth of okra plants, and 15% showed maximum growth. In case of spinach, 15% vermicompost shows a maximum growth of the spinach plant. In case of wheat, 25% and 25% and 15% show the seedling and vegetative growth of wheat. And 20 to 35% of vermicompost concentration shows the maximum seed germination and vegetative growth of the barley plant. After this, why we are doing the isolation of microbes from the Yersinia foetida gut? Because these microbes are involved in the degradation of solid waste material into the vermicompost, and then these are also bacteria used as a plant growth promoting bacteria that is involved in the also present in the vermicompost. So we did isolation and identification of vermibacteria from the Yersinia foetida gut, because we use Yersinia foetida for the vermicompost production, and different methods and techniques were used. Text staining, morphological, cultural media were used. We screened the plant growth promoting properties. We also screened the hydrolytic enzyme productions, and then molecular identification was done. Here is the outcomes of objective A, isolation and culturing techniques and staining techniques. Outcomes of B, that different enzymes produced, hydrolytic enzymes produced by the vermibacteria. And here is the plant growth promoting traits. And here is the over tabular form of the complete biochemical characterization of the isolated bacteria strains. After this, we did molecular analysis by using ribotyping, and we did blast analysis, and phylogenetic tree were done. And then after this, we submitted the sequences to gene bank. These are the blast analysis, results of blast analysis. Phylogenetic relationship show that these microbes are related to Bacillus mycoides, Pristia megaterium, Staphylococcus humanis, Bacillus mycoides, Bacillus licheniformis, Bacillus subtilis, Bacillus mojavensis, Bacillus mycoides, Bacillus thionensis, Bacillus anthracis, and Bacillus paranthracis. And these all vermibacteria were identified, and then their sequences are submitted to the gene bank, and gene bank allotted these section numbers. After isolation of vermibacteria and the production of vermicompost, we also check the impact of integrated management system. The combination of vermicompost and vermibacteria on the seed germination of the ornamental plants. And we observed that when we used vermicompost and vermibacteria in combination, they show the maximum growth of the pansy plants and the marigold. Here I am also clearly show the length of the whole plant of the marigold, and here is the pansy plant, and the growth whole plant length and growth of the plant when use the integrated management method. What comes out from the impact of integrated management system on ornamental plants, that vermicompost, we know that they are the phosphate solubilizers. They augmentation the plant growth because they are rich in nitrogen, phosphorus, and potassium. They improve the pH and electrical conductivity of the soil. And when on the other hand, plant growth promoting vermibacteria enhance the seed germination and vegetative growth parameters of the plant, and enhance the nutrient uptake, and also fix the nitrogen, because they have a potential plant growth promoting traits. On the other hand, beside these, the plant growth promoting bacteria not only involved in the to enhance the plant growth and the plant development, but also involved in the remediation of heavy metals. As we discussed, that vermibacteria, vermicompost, vermibacteria are used as a microbial biofertilizers, and these vermibacteria also used as a biodegrader and also used as a vector remediator. So how they work? Here is a summary of the work, that plant growth promoting bacteria involved in the nutrient uptakes, improve soil health, promote seed germination, enhance the seedling parameters, biocontrol, antimicrobial, involved in the phytostabilization, phytoextraction, phytostimulation, and rhizofiltration. Because due to the presence of of these plant growth promoting traits. On the other hand, as a vector remediator, they remediate the chromium, cadmium, and lead. And due to presence of different efflux transporters, metal binding proteins, operons, surface protons, metal chelating agents, and plant growth promoting traits. And due to presence of these activities or these traits, earthworms or vermibacteria detoxify the toxic material into non-toxicity due to different mechanisms, either biotransformation, bioaccumulation, biosorption, oxidation and reduction, detoxification, methylation and demethylation, and expulsion of the heavy metals. So here is the summary of my talk, that vermicomposting offers a wide range of benefits, making it an environmental friendly and sustainable method for organic waste management and soil improvement. Earthworms and their metabolic products, either in the form of vermibacteria, in the form of vermicompost, may work as a driving force in the sustainable food production, where improving the soil health and fertility. Agrochemicals could be replaced with a sustainable development technology, vermicompost utilization, because vermicompost is cost-effective, highly porous, and NPK-rich product for nursery raising of ornamental plants and the crops in the Pakistan. The integrated management system showed great impact on the germination of ornamental plants. Vermibacteria found in earthworms, gut, and in vermicompost play a role in the solid waste degradation. So we can say that vermibacteria could be used as a potential source of biofertilizer, biodegrader, and vector remediators. And overall, the vermicompost are warm costing, and the vermibacteria are valuable fertilizers that could be used to enrich the soil and promote plant growth, as well as could be used for the degradation of solid waste material. This whole work, our work is published in different international journals. Pakistan Journal of Zoology, in PLOS One, Journal of Ole Science, in Helion, and PLOS One. Thank you for the